I'm Daniela Bittencourt, and I am a researcher from Embrapa in Brazil, and also a collaborator, a Jones collaborator, mostly focused on this task in order to develop synthetic cells from non-living parts. And actually, I will present to you on work in progress of what we are doing in trying to reach this goal. So, oh, I'm not sharing yet, sorry. Okay, so many of us in the synthetic biology field, I believe that the major goal for synthetic biology is are trying to develop a synthetic cell through a bottom-up strategy. So, but why we need that? So many of our other colleagues talked about this already, but just saying a little bit about it. So it would be nice if you could design a specific cell to mimic behaviors and functions that we need, that we aim for a specific task. Also, the synthetic cell can have properties and components that are not present in the living system and might be useful for some application in medicine or agriculture, for instance. And of course, this is a way that we could have full control over the functional and structural properties of a cell. And of course, try to understand how biological cells work perfectly. But as Tyler was presenting the last presentation, he was talking about his efforts and strategies in order to develop a synthetic membrane. Well, for me, this is actually the first part. I think we need to think about it. How to develop a membrane that will be capable of receiving a new genome and boot up to a new living cell. Well, this is a very, very difficult task once the membrane has different functions and are very essential for a cell for its life and to, how can I say, to be considered actually an organism, a life organism. The membrane is completely essential. So it has different functions that selectively permeable. They have to have different proteins that will manage the bidirectional flow of ions and molecules, create temporary gradients, interactions with cells and if their environment. And of course, it has to be capable to change shapes. Different kinds of materials have been used trying to mimic the cell membrane and, of course, its functions. However, why don't we take advantage of their own cell membrane? In this case, thinking about mycoplasmas and the development of the JCVI-SIN1 and SIN3 cells with synthetic genomes and their capability of the mycoplasma membrane to receive these genomes, why don't you just try to use their own membrane? So this is what I'm trying to do. I recently work described the generation of SIN cells, which is the name for simple cell. And actually, these cells, they are, or we also call them as zombie cells. They are cells that do not have genomic DNA inside of it. Actually, what they do was insert a construct with an endonuclease called ICO, which is capable to recognize the sequence present in the RRL gene of the 23S rRNA. Depending on the number of copies of these genes in the cell, the degradation of the genome of that cell goes faster and it's more efficient. So this strategy has been done for 3 different species, one of them which is E. coli. But since the endonuclease mediated double-stranded breaks in the genomic DNA of the cell, it also kills the cell, of course. And if you do not have a very controlled expression of the endonuclease, well, this is very difficult to get a cell, let's say an empty cell without the genome, but that continues being functional at least for a while. So in order to acquire that, what we did was to develop a genetic circuit where this endonuclease actually is under the control of serine integrase that were described from our group a couple of years ago. So what we did was to construct a genetic circuit where the integrase is under the control of the tetracycline promoter. And once it is expressed, it recognizes the endonuclease gene by these two specific sequences, which we call ATTB and ATTP, and it is capable to reverse and actually put in the correct direction for gene expression. So that's what we did with our endonuclease. So this blue arrow would be the endonuclease gene, and after the integrase activity and twisting the gene, it goes in the correct direction. Here we have our promoter, which is going to be a constitutive promoter. We use this spirulina promoter, and then the endonuclease express, and we hope that we have genome degradation. So what we did then was to transform the JCVI SYN3B and SYN1. We introduced this genetic circuit into the lengthy pad that is present in their genome. So we have these circuits integrated in their genome. So hopefully what we expect is once we have the tetracycline induction for the integrase activity, and once the integrase is active, we have then the endonuclease activity integrating the genome, and finally we're going to have an empty bag, or our zombie cell, stem cell, whatever. And this actually empty bag, or it can't be considered as a living cell anymore. It will be capable of being capable of having some activities for a while, but then in a couple of days, it will be gone for like no activity at all. So, but for at least for during this small period of time, we believe that we can manage to insert another genome inside it and then put up a living cell, or simply another construct, a simpler construct, which we can make the cell viable for a while in order to develop a specific function, or even study specific metabolic pathway without interference of the host genome. So another thing that we did also was to include the arginine, the ADI metabolic pathway into our cell. Once we inserted the genetic circuit inside the JCVI SYN1 and SYN3 genome, we also transformed those cells with this metabolic pathway, which was well introduced by our colleague Bert Coleman last week's presentation during the workshop. So this pathway is used by some species of mycoplasmas as an alternative pathway to produce ATP, but it's also very easy to follow its functionality, because it changes once the arginine is degraded into ammonia and CO2. It has a change on pH on the medium, so there is a biochemical assay that we can perform in order to identify its activity. So this is not only an alternative to have maybe more ATP, although now I'm not very sure about Pullman representation like last week, but okay, we can work on that. But this will be, with this pathway, we'll be able to follow for how long our zombie cell will be active, or have our the cytoplasm will be active for how long. Like, okay, it's going to be active for 1, 2, 3 days, or even a week before genome degradation. So here we started, once we have transformed the cells, the first thing that we did is to check if our ADI pathway was functional in our cell. And this is like, we can see the measurement of citrulline release from the enzymes activity. And here we have our clone with, we call it K2R5. It's just a name for the clone, one of the ones that we acquired. And we compared with our strain, SYN3B strain, and this is Mycoplasma capricolum, which has this pathway naturally in their genome. So, okay, this is functional. And now we move forward in order to check the viability and the functionality of our design in T9 ICU genetic circuit. So then we performed a quantitative PCR where we amplified the region of the the region that the endonuclease targets in the RRL gene. So we design primers flanking this region. And here what we see is the average number of cycles required for the amplification of the target gene, this target region where the endonuclease cuts. Well, we can see that this is for induced. We have 3 clones here. Oh, I'm sorry, which is the same clone, but 3 triplicates. So here we have non-induced, and with that, we have the induced ones for the expression of the endonuclease. So here what we see is that we need a larger number of cycles to see the amplification of the target gene. So this shows that we have lower a decrease in abundance of the target gene in cells with degraded genome, showing that actually the degradation is happening. So here we have the same analysis in 24 hours after induction, and here we have at 48 hours after induction. We also performed this quantitative PCR in earlier stage, but at 24 hours is the time of incubation that we needed to get the most efficiency of our system in degrading the genome. So here, this is very interesting. We have a movie, it's a time lapse that we did, showing the degradation of the genome DNA through time. So these are thin 3 cells, stained, actually I believe this video is from scene 1, stained with DEPI, and it was prepared in agar slides in the presence of tetracycline. I'll just put it again. So we can see the cells like the DEPI just going down. Here we have a graphic showing the fluorescence going down, and here we go up to 20 hours. And that's why showing that our qPCR analysis, the timing of the degradation of the DNA is very similar to what we acquire from quantitative PCR. So, but this is interesting because when we perform color change units with our clones, what we see actually is that after 24 hours, okay, we don't see these are SYN3B, our negative control. We also have the integrase controlling MHRA expression. This is just to see that the integrase is not interfering anything in the genome or the viability of the cells. So we have our controls. After 24 hours, you can see some growth from the dilutions, but no growth for our cells with the endonuclease present in their genome. However, after 48 hours, we can see some growth. So we have some cells that are surviving from our, well, strategy. The idea, actually, no cell surviving, but anyway, okay, we still have some. So we decided to perform flow cytometer analysis in order to identify exactly which is the efficiency of our system in degrading the genome. And what we did was staining the cell membranes with FM143 dye and also the genome with DEPI, and trying to check for, here we have double positives, would be very healthy and good cells. We have membrane, we have genome DNA. Here we only have cells. We have cells only with DEPI, no FM. Here we have the membranes, which would be our Zumbi cells, the cells that we are targeting, the ones that there is no genome DNA. And here probably double negatives, the ones that probably will be dying or the cell debris and stuff. So don't want this part. So here, what we did after this strategy, we check for the genome degradation through time. So we check for 6 hours, 12 hours, and 24 hours, if I'm not mistaken. So here, if you see, So here, if you see, we have non-induced and induced ones. We work with 3 different clones, and we have SYN3B as our negative control. So double positive cells are the cells that presenting DEPI and FM fluorescence. So here we have similar results, although after 6 hours, we can see a lower number of double positive cells in our treated cells. And here, if you see the percentage of the cells, the induced ones, the percentage of only FM positive cells are much higher than the non-induced ones. And this same pattern we can see after 12 hours, which we have higher amount of cells with only FM positive, which are only FM positive. And after 24 hours, we reach the maximum amount of it. So, but what we see here is that we have in the best clone here, we only have 40% of cells FM positive. So it means that we still have almost 6%, 60% of double positives. So these guys here somehow are being capable to escape from my trick. So this is, I don't want to happen. I want actually to increase the efficiency of the system. And interestingly, oh, here, okay, just like to see the kinetics. So here it's a better way to go. Then we can compare all of them to the time. So if you see here, this is the amount of cells, and here we have the percentage showing the 40% efficiency of genome degradation. And here we still have 50 to 60% of intact or parental cells present in the population. So, but interesting is when we work with SIM1, what happens is that SIM1 starts dying. Somehow the endonuclease activity is really interfere on the viability of the cells. And actually we have a higher number of double negative cells through time here. So here we can see the fluorescence of the epi going down. This, the red one are the treated ones, the induced ones. But also we do have a lower fluorescence of FM, which we didn't want to. So it seems that we are having cells that are dying and they are disrupting. Differently, what we see from the SIM3. So those data that I showed to you, we acquire using the JCVI SIM3V as our chassis in order to develop the SIM cells or zombie cells. But here we can see that JCVI SIM3, it works perfectly differently from JCVI SIM1. Well, probably because of the amount of genes, probably have more genes that are defending themselves somehow. So this is something that we need to work out to look it up for better for this and see what exactly is going on the metabolism of the cells. This is progress of what we have so far. So what we can conclude is that, well, our genetic circuit is functional and it's time dependent, reaching into maximum genome degradation at 24 hours after incubation, and so far has an efficiency of 40% in JCVI SIM2V cell population. So we still have 6% of the parent cell there surviving with their genome intact. So what we're going to do next? Well, we need to better understand the ICU impact on JCVI SIM cells metabolism, need to improve purification methods. Oh yes, this is something that we tried. So, well, since we still have some parental survivals, some cells that are still with their genome, we need to purify those populations. So we are starting performing cell sorting using also the flow cytometer. Unfortunately, we tried twice so far, but we are acquiring some contaminations, and after 3 days, 4 days of incubation, we see cells growing and dividing where it should be only zombie cells. So we need to improve this method. We have other strategies in mind to perform. And then after that, we'll be capable to evaluate the mycoplasmas in cell longevity, for how long they will be functional, so we can then perform the genome transplantation analysis and try to insert the whole genome inside of this membrane that now that is just the membrane, like say an empty sac, and then see if we can boot up a living cell. So with that, I finished my presentation. I thank you everybody that somehow helped in this work, collaborate on this, all the advices from John, my friends at JCVI, Sage, Mark Novati, Kim Wise, miss you guys a lot. So hope to see you soon. Thank you very much.
Very nice, Danny. So I like that you're trying to solve some of our genome transplantation problems with a truly novel approach. Questions for Danny?
Daniela, this is James Dobbins Peck
Hi.
in Birmingham, Alabama. How
Hi,
you doing?
James.
Enjoyed your
I'm fine, and
presentation.
you? Thank
One
you.
quick question. I was wondering what you were planning to do to protect your transplanted genome from the nuclease that's left over from degrading the host
Hmm.
genome. Do y'all have
So
plans to
yeah,
deal with that?
yeah. Well, this is something that we have. There are some gaps there that we still need to analyze. So the first strategy actually was to insert all the constructions together in one plasmid with Mycoplasma origin. So the way they would be functional there after genome degradation. But then I realized, okay, but if you have that, the internuclease will be expressed all the time, and I won't be capable of receiving another genome. So that's why we decided to introduce it inside the genome of the minimal cell, because once it worked and degraded the genome, it wouldn't be produced anymore. However, we will still have some of this enzyme present in the cell. So this needs to be evaluated. So the idea is that when we'll be capable to purify our population of stem cells, we are going to perform RT-PCR, different analysis to check for how long this enzyme is present, and also try to perform the genome transplantation process in different time frames, after 24, 48, 72 hours after genome degradation, and see if we can be successful on that.
Is there any way you can engineer, and I don't know, I'd have to spend a couple of months in the literature, I guess. Is there any way we can engineer a increase in the half-life or a decrease in the half-life of that particular
Well,
nuclease?
yeah, maybe you can insert some region for that will be recognized for some enzyme for degradation, faster degradation.
Yeah,
Maybe possible.
yeah. It's
Yeah,
very nice work, Danielle.
something
Good
that.
talking to you again.
No, nice talking to you too, John. Thank you. Thank you for your tips.